Our topic today is development of an AI-powered prediction of BRAF mutation and patient response to immunotherapy from H&E stained slides in metastatic melanoma cancer. I'm Thomas Colarusso, CEO and co-founder of BioAI Health. Our company is focused on developing novel AI-powered precision medicine tests for clinical trials. Our technology automates screening and selection to improve patient outcomes. Today's presentation outlines a use case that was developed in tandem with one of our strategic academic cancer center partners, Dr. Paolo Ascierto, and the Institute of National Tumor Foundation, Pascale in Naples, Italy. BioAI offers a portfolio of AI solutions and services. We continue to expand our capabilities to support a broad range of disease areas, biomarkers, and various drug therapies. Our agenda today is, number one, we'll introduce the speakers, and then we'll cover current challenges in clinical oncology, and then the rationale for development of AI predictive tests, and then an overview of predictive model development from BioAI Health. And then we'll have a QA session, and I encourage each of you to submit questions during the webinar and the chat, and we'll address them at the end of the meeting, at the end of the presentation. So I'm pleased to introduce our first guest speaker today, Dr. Paolo Ascierto, a practicing medical oncologist and world authority in the field of immunotherapy. Paolo is the director of Department of Melanoma, Cancer Immunotherapy and Development Therapeutics at Institute National Tumor Foundation, Pascale in Naples, Italy. Paolo is a scientific reviewer for leading medical journals and sits on several medical advisory boards and committees. He has presided as PI on over 150 clinical trials, and he has authored more than 500 publications in peer-reviewed journals. Paolo will provide an overview of current challenges in clinical oncology. I'm also happy to introduce our second guest speaker, Alan Gerald-Salmi, our Chief Services Officer and co-founder for BioAI Health. Alan has an extensive background in life sciences and the pharma industry. We have been working together to build the company over the past 2 years. He will provide an overview of the collaboration with Dr. Ascierto and highlight project design, biotechnology, BioAI Health's technology stack, and review applications that we have developed and corresponding results. Paolo, the floor is yours.
Thank you so much, Thomas. And first of all, I'd like to thank you as a BioAI Health and the organizer for this kind of invitation. It's a really pleasure for me to be here today to discuss about the need of biomarker and why this is particularly important in the field of melanoma. Thank you for the slide. So, as you know, melanoma is one of the cancer that the incidence is increasing, because every year we have more patients with the melanoma, and this it's important because the melanoma was a rare disease several years ago, but now it's one of the most important cancer as a number. And what is particularly important for melanoma is the complexity of this disease, because we have several kinds of melanoma. Consider the uveal melanoma, mucosal melanoma, behind the cutaneous melanoma, that from a biological point of view is different. And this is the reason because we are doing a lot of translational research and basic research with a lot of genomic data, because we want really to characterize the tumor, because what we need is to be able to have a personalized approach to this disease. The next, please. Just in order to give you some numbers, in order to make more clear about what we are discussing today, the number of the new melanoma that globally were diagnosed in 2020 was more than 300,000. And as I told you, in the majority of cases, this melanoma, these 300,000 more and more, fortunately, in the majority of cases are thin melanoma. But our concern is about the thick melanoma with the possibility to have a metastatic disease in a high percentage. What is important is that in the last years, the history of melanoma was changed thanks to some new compounds like the BRAF and MEK inhibitor, and in the field of immunotherapy, thanks to the anti-CTLA-4 and anti-PD-1, like nivolumab and pembrolizumab, who changed the history of melanoma, as we will discuss very soon. And what is important is that thanks to the activation of immunosystems, we are able to cure some of these melanoma. And you will see some curves. The next, please. What we have seen with anti-CTLA-4 and anti-PD-1 is that we have patients with the long-term benefit. As I told you, we can cure some patients. At the beginning of the history with checkpoint inhibitors, ipilimumab was seen to be able to get a long-term benefit for the 20% of patients. With anti-PD-1 nivolumab, we have seen that this percentage was higher, 40%. Now we know with the combination with ipilimumab and nivolumab that at 6.5 years, we are more than 50% of patients still alive. So this means that we are able today to cure more than 50% of metastatic melanoma. And this is, of course, an important news for our patients. Consider that before the 2011, when we had just chemotherapy, only the median overall survival was 6-9 months, and only the 25% of patients was alive at 1 year, were alive at 1 year. And at 2 years, almost all the patients died. Now we have these results with this compound, and as you can see in the slides, we have over 760 ongoing clinical trial for the metastatic melanoma. The next, what we learned during the past year from the 2011 so far, when we had the approval of some of these compounds, and year after year we added some compounds for our patients, we knew and we learned that the patients who are in the tail of the curve, as you can see in the left of this slide, are the patients with good prognostic factor. That means the patients with normal LDH, with the low tumor burden. And we know also from some study with the combination of ipilimumab and nivolumab, but even patients with brain metastases, if these brain metastases are asymptomatic, are small and few, even these patients could be in the tail of disease. So the patients with good prognostic factor, but these factors are clinical factors. We don't have biomarker. We have just clinical factor that can help us. All the other patients with elevated LDH, with more than 2 and big brain metastases, with high tumor burden, with several metastases, are in the black area of this curve, so continue to die. For these patients, we need to do more. But at the end of the day, we need biomarker to characterize these patients. In other things, if the good patients are the patients who can get the long-term benefit, the translation from metastatic disease to adjuvant next was the next step. And we know today that this agent, target agent, even BRAF and MEK inhibitor and anti-PD-1s, are effective even as adjuvant, so after the resection, in order to prevent metastasis. And this is another important news. But even in this field, there is about 50% of patients that can get a benefit. The other 50% continue to relapse. And even in such case, we need to do more. The next. So coming back to the possibility to get cured, the 50% of patients, as I told you, If it's true that we can cure the 50% of metastatic melanoma patients, there is another 50% that continue to die. The 30% of these continue to die within the first year still. And it's important at the moment to predict where the patients in detail. We have just clinical factor, but as I told you, we need even biomarkers. And this needs is important also because in the next future, we could have several possibilities, several treatments that we should know where the patients that can get a benefit from one treatment or the other treatment. The next, please. Because if we consider these 50% of patients that continue to die, probably the key is in the tumor microenvironment. What does it mean? It means that for the 50% of patients that can get a benefit in the field of melanoma, but you know that immunotherapy is also important in other kinds of cancer. We are talking today about melanoma, but consider the other cancer where immunotherapy may be still good, but probably the percentage of patients who continue to relapse and die are even more. The key is in the tumor microenvironment. So if I have the so-called inflamed tumor microenvironment with a high number of lymphocyte, immunosystem cells, this probably is the kind of tumor microenvironment of the patients who are in the tail of the curve. In the other patients, probably we have the so-called immunodesert tumor without any immunosystem cell, or the immune-excluded tumor, where the stroma may affect the possibility from immunosystem cells to kill the tumoral cells. So we should be able now, with new drug, to make immunodesert or immune-excluded tumor inflamed in order to make this tumor responsive to immunotherapy. The next, one of the strategy is the combination approach. Combination, so immunotherapy can be combined with all the other tools that we have at the moment available against cancer. Chemotherapy, and we know that in the field of lung cancer, the chemotherapy plus immunotherapy is one of the strategies. We can have radiotherapy that can also synergize with immunotherapy and make the tumor more responsive. We can have the combination of immunotherapy with target therapy. In the kidney cancer, you know that we have some target agent, anti-VEGF, that can synergize and increase the efficacy of immunotherapy. But we can also combine immunotherapy with other immunotherapy. So checkpoint inhibitor and checkpoint inhibitor. We know that ipilimumab and nivolumab is more potent than the single agent. And if we look to some of this combination, the next. Last year, it was presented the results of the Relativity 047 study. The combination of nivolumab, an anti-PD-1, with relatlimab, an anti-LAG-3, an additional checkpoint inhibitor that was compared to the monotherapy with nivolumab, with progression-free survival as main endpoint. And as you can see in the next slide, the study was positive. So this overall survival, progression-free survival, is not from the relativity. This is from the Checkmate 066, that is this trial which compared nivolumab and dacarbazine. Sorry for the confusion. But in the Relativity 047, the combination nivolumab and LAG-3 was better compared to the monotherapy. And FDA approved this new combination a few days ago, as probably you know from the press release from FDA. The next, please. So having said that, we know that now in the field of melanoma, we have treatments like immunotherapy that can cure the patients. Can cure the patients with metastatic disease. But at the moment, we don't have a biomarker. We cannot select the patients who can have a benefit from one treatment and another one. Of course, we strongly need this because now we have an additional combination that can be useful for our patients, and we should know what are the patients who can get a benefit from ipi-nivo or from nivo-rela, even because the first treatment have a higher toxicity compared to the second treatment. So if we will be able to find biomarker, of course, we will be able to choose the best treatment with also the best safety profile for the patients. We know also that there are some other markers that can affect the response to immunotherapy, can also be important, as I told you, for the toxicity. But at the end of the day, this biomarker will be useful in order to start what we will probably see in the next future, the personalized immunotherapy. And with all this opportunity, it's something that we will see. Having said that, I'd like to thank you for your attention. At the end, we will have the Q&A. And now is my pleasure to introduce Dr. Ala Jerusalem for the next speech. Thank you, Alan.
Thank you. Thanks, Paolo. Appreciate the introduction. So first of all, thanks everyone again for joining the webinar. And during my talk, I'll focus mainly on the technical aspects of the project and address the workflow that we used together with the results that were generated from our models. But before that, just a quick background on the work. So everything that you see here is the result of an ongoing collaboration between BioAI and the Institute at Naples, where we have a very strong alliance with Dr. Paolo Asciorto's lab to explore the potential of using AI-powered tests on clinical data for metastatic melanoma treated patients. In this collaboration, Dr. Asciorto's lab provided the image data, the clinical metadata, and the patient response data. And then BioAI had its pathology team conduct all of the image QC needed. All of the annotations were done by our team as well. And then our pathology team supervised and worked closely with our data team in the development of the predictive models that I'll go through. That included the tumor detection module, the BRAF expression model, and as well as the patient response model. So BioAI PredictX workflow has been designed leveraging all of the years of experience that we have in building these automated image analysis tools, always with the goal of having an end-to-end automated solution that can work across an entire data set. So everything starts with the selection of the tumor-specific region within the tissue. And here we accomplish that by a tumor segmentation model that's very specific for the tumor type of interest. On this case today will be melanoma. So as you know, we're dealing with very large images, and for computational purposes, these images need to be broken down into tiles. So what our tumor segmentation does is split down all the tissue into three main compartments. So we have the background that gets excluded, the non-tumor region that typically gets excluded as well, and then finally the tumor regions. Of course, if we need to, we can also incorporate further grouping here. There's been projects where we need to look at tumor microenvironment as well, as opposed to just strictly tumor, non-tumor regions. So once we have the tumor tiles generated, we then perform an automated image QC where we exclude any bad tile images from the mix. And this is typically because they can have an adverse effect on the model development. And then finally, we can split the tiles into our desired groups using our predictive models. So today we're using two separate binary models, one of them looking at BRAF molecular expression, and the other one is looking at patient response to treatment. However, we do have some projects where we use the same workflow, but as opposed to being a binary model at the end, we're looking at a multi-class classification model. So in terms of the data set that we use for the project, we started off with about 92 images, out of which 84 images had a BRAF designation on them, and 65 of those images had also been treated with a PD-1 inhibitor. Within the BRAF data set, we had 51 normal cases and 38 cases that had a mutation. And for the PD-1 treated response data, we had 49 patients that did not survive drug treatment and 22 survivors. So as you can see, once we create the tumor tiles from those corresponding cases, we're left with just over 1 million tiles overall for the BRAF model. And for the response data model, we had roughly 850,000 tiles. And then what we do is we do a very typical split where we used a 60-20-20 split between our training group, our validation group, and our test group. And just to reiterate, the test set is a held back data. It never gets exposed to the model during training, and we only use it to assess the model performance. And that's the data that I'll show towards the end of the presentation. So in terms of the developmental steps that we had for the tumor generation tile, so we did not have to build a melanoma tumor segmentation from scratch. What we did is we used transfer learning from a previously developed tumor model that BioAI had developed on lung non-small cell, and we optimized that for the melanoma project. So this was done by incorporating hundreds of pathology annotations, which covered both tumor and non-tumor regions. And we do that in an iterative process on a collaboration between our pathology and our data science team. And typically we'll go through these iterations until we have a fairly good AUC. Typically we're looking for 0.8 or greater. And then at the final step, we do have a pathology team do a qualitative assessment on all the tumor regions to make sure that we're not missing anything and the segmentation is good. So once we're happy with that, then we can move forward. So the next slide shows a few representative images of what these heat maps look like of this tumor overlay. So here in the image, you can see in blue the tumor region and in green the non-tumor regions as were extracted from these images. So if we look at the overall data that we only had 2 images where we had just under 100 tiles. We had about 90 and high 80s for those 2 cases. However, the majority of those images, we did have quite a lot of tiles, the bulk of them having more than 10,000 tiles per image. So one area that gets overlooked a lot of times when these types of analysis is done is on the image QC. The typical workflow that most people do is doing a QC prior to the start of the analysis, where they may exclude the entire image. However, as we all know, once we look at the image and the higher magnifications, you can always find some areas that could have an impact on your model performance. And I'm talking about areas, and you can see some examples here, areas that may have small tissue folds, or if you have some areas that could be a tile full of red blood cells, you may have the tissue pigmentation that you get, or just any other artifact. And those could be an issue for your model development. And then again, when you're running your model in the end for the testing. So what we do at BioAI, we always run these tile images through our QC model that we developed, and we always optimize that model for the specific tissue of interest. So for this project, we took our QC model and we optimized it for melanoma. And we do that just by running through thousands of tiles that have been labeled for the good and the bad regions. On this case, we had an AUC of 0.86, so it's fairly accurate at detecting these bad areas. And what we typically see is roughly 3 to 5% of tiles tend to be excluded after we run through this model. Okay, so as I mentioned earlier on the talk, we developed a probability model with the goal of being able to predict the BRAF status from a patient looking only at the H&E image. So to reiterate, we had 84 cases that had the BRAF status, where we had roughly 800,000 tiles out of the images that were used for training and validating our model. So the data split for this particular model was fairly good. We had a 60-40 split. And again, for those of you not familiar with building these deep learning models, we always aim to have a 50-50 split. But since we're typically dealing with clinical data, that's usually not the case. But 60-40 was a fairly good split here for the BRAF model. So if we focus on the performance of our test set and explore those results, the result set contained roughly 315,000 tiles. And the results came out fairly good in terms of accuracy of how the model performed. Again, this is an initial model. We have not optimized the model yet. This is something we will do at a later stage, though we have extremely encouraging results from what you can see here. For those of you not familiar with some of these terminologies that we use to measure the performance of the model, So when we talk about precision, we're referring to what proportion of the positive identifications were actually correct. And when we look at the recall measure, we're looking at the proportion of actual positives that have been correctly identified. And then the F-score becomes the harmonic mean between the precision and the recall. And you can see here we had between 70 and 80 on some cases, so fairly good numbers to work with. When we look at the AUC for this model, it was 0.75. And again, as a reminder, the AUC refers to the ability of our classifier to properly distinguish between the two classes. And the AUC is also illustrated on the ROC curve here at the bottom of your slide. One thing to mention, some of you may be wondering why the curve looks a little bit different. Typically, you see a nice AUC curve. But what we did here is we took a single point when we extracted the data from the predictions, and that's why it kind of looks a little bit differently. And then at the bottom left, we're showing you the case level performances. So on the top right is tile by tile level. And then when we convert that into a case levels, we're still getting those 70% or 80 percentile accuracy. So fairly good results to start with for the BRAF model. It really does indicate quite a lot of potential to explore this further. So for the survival model, our goal here was to determine if we could, using only the pre-treatment biopsy image, predict the response to therapy, which again, in this case, was a PD-1 inhibitor. And using a very similar methodology, slightly different architectures, but similar methodology from what we used before on the BRAF model, we went ahead and did this work. As you can recall, the number of cases where we had that had been treated with the PD-1 were slightly less than what we had for the BRAF cases. But in terms of tiles, we still had quite a bit. We had 800,000 tiles available for the training and validation of the model. The split here wasn't quite as good as before. Before, remember, we had that 60-40 split. For this particular model, we had a 70-30 split, which we can still work with, but it's a little bit more challenging since it's not that 50-50 that we're looking for. However, when we look at the test results for the survival model, the numbers, not only they look fairly in line with the previous model, but we actually saw a slightly better performance here. And again, using the same metrics that we used before on the precision, recall, and F-score, we can see numbers on the high 70s and 80s percentile. The AUC was a little bit better than what we saw before. Before we saw the 0.75, now we're getting an AUC of 0.83. Though again, this is initial results. We would like to now further train the model and optimize it to make sure that we can keep the same level of performance that we see here. But super encouraging data. We're very happy to continue this collaboration and explore this further. So just to summarize the workflow that we used, again, to highlight, everything starts off with a single H&E image. Again, the goal is always to be able to predict all of these mutations, all of these treatments, looking at a single H&E image. So again, what we do is we take the H&E image, it undergoes through our tumor segmentation model, where we select for very specific tumor regions, and then we can create our tiles and run through the QC module to remove anything that shouldn't be there. And then once we're left with this nice clean images, we can run that through our predictive model, which on this case here could be either looking for a specific mutation or looking at a response to therapy. Just to highlight, we have successfully applied the same methodology and workflow to numerous other projects using different tumor types, using different molecular markers. However, the workflow is fairly consistent. And one thing to notice is that as we're working with these clinical images, the images aren't always coming from one clinical site. So there's been numerous cases where we had images coming from multiple clinical sites, sometimes even coming from different scanners. And we have been able to account and develop methodology that we can incorporate all of these different images from sites into a single workflow. So the ongoing effort that we have now is to go a little bit beyond the actual predictive models and really start getting an understanding of what are the features that are responsible for what we see here, for that classification between BRAF positive and negative, or between the drug response. So at BioAI, we employ a deep learning visualizer that allows us to do that. And you can see an example of the visualizer here at the bottom. And what the visualizer is doing is really showing the performance of our model as a graphic representation on the tissue. However, as you can imagine, we're going through 700,000, 800,000. So we're going through quite a lot of tiles here. So the amount of data that this generates is in the millions of data points to analyze. So it becomes a fairly complex workflow. So what we're doing now is we're in the process of employing a high dimensional data analysis where we can condense all that information down to a few key meaningful features that will shed some light into the tissue regarding where that signal is coming from. And if we just spend a quick minute to go back a slide, if we just spend a quick minute here to look at this example, if we zoom in on the image, you can see that all of these tumor nuclei have sort of a halo around it, which it indicates there's something happening on the surface of those nuclei, which is driving the behavior of the model. And this is what we aim to explore as we continue this collaboration to really understand where the BRAF signal is coming from. Okay, we can move on. So finally, as a next step, so what are we currently doing? So we're working with Paolo to obtain additional samples so we can go back and further optimize and then validate the work that we did. And then we would also like to explore the potential of combining the models, right? So as opposed to looking at one model for BRAF and one model for the survival, what happens when we look at the response signal in patients that had the BRAF mutation? So those are some of the combinations that we aim to explore once we continue and get a few additional images to work with. So with that, I would like to thank everyone involved in this work. So a special thank you to you, Paolo and Gabriel at your lab that have been tremendous help in getting us the images and regularly meeting to discuss the project and the progress and what the next steps are. On the BioAI side, I would like to point out Krishna and Zinging, who've been crucial at running all of these models that you saw today. And also Robert, who's been our pathologist, working very closely with us for quite some time now. Thank you, everyone, for your time today. We're happy to take any questions.
Well, thank you, Alan, and thank you, Paolo, and thank you, everyone, for attending. And I will open the floor up to some questions that we received. And the first one that came in, I think this would go to you, Paolo. It says, what are the advantages in making a prediction on H&E stain slides from patient biopsy or full surgical resection?
So, important question, because to have an evaluation H&E, it's important to optimize the plan for the treatment of patients, because I can save some time, and for the metastatic, mainly for the metastatic disease, the time it's important to start the treatment as soon as possible. Consider so the patients, there are some patients, just for an example, with no primary lesion, with just a biopsy for a metastatic disease. And in this biopsy, in an H&E, you can have an evaluation of BRAF without wait for the 7-10 days that in general we need for the BRAF mutation. That these 7-10 days may be important when you have a symptomatic disease. So to have an evaluation H&E is always better than not.
Very good, thank you. Another question.
Just to follow up on that, it's important to realize that the H&E slides are already available. And the models that we're running, that entire workflow that I showed you, that can be run in under a minute. So it's very quick to go through these H&E images. So there's a lot of additional benefits that go tandem to what Paolo just said.
Awesome. Another question came in, which is, can you run the 4 models concurrently for deep learning?
Yes, we can. So I kind of broke down that workflow doing one model at a time. However, as we're running through this, like I just said, in about a minute, everything goes from start to end. So it's one single process running the entire images.
The next question, I think, goes to both of you, and it says, what will be required by BioAI and Institute in Naples to transform this prototype application into an LDT, a lab developed test, or an IVD. So how would you guys tackle that?
Yeah.
Alan is
So I'll get started. So from our perspective, I think we need additional data, right? Once we have more data in our books that we can do a round of optimization, and then getting a separate data set and validating against that data set would allow us to then transfer this into a clinical setting where it can go through and be run clinically. Of course, it has to go through the needed rigorous validation process. We do everything under design control, so we have all of that recorded. So once the model is validated, then it's ready to move forward. Paulo, any additional?
Yes, of course. So as first, as you said, we need more samples for an internal validation. This was just the first phase. And of course, we need to do this on more and more samples. And after this, we need like an external validation as for all the other biomarkers involved other centers with other samples, more centers. And then when all will be ready, this probably maybe this test may be translated in clinical practice. But validation is of course fundamental when we talk about biomarkers.
Awesome. No, that's a great response. We have another question that says, can these models be transferred into new tumor indications like lung cancer or breast cancer? So you talked a lot about melanoma. How could you move this over into another tumor type that we haven't seen yet?
I believe this is fundamental. Before to give the words to the floor to Alan, mainly for the lung cancer, because in lung cancer we have several mutations that can be targeted by specific drug. And for this reason, will be more and more important for all those cancer where we have several possibilities with an H&E. As Alan said, in 1 minute, you can have, of course, all the possible marker for the specific cancer. And Alan, the comment from you and the question from my side, because looking to the lung cancer, differently from melanoma, we have sometimes some, small biopsy or the cytologic assay. In the next future, do you believe that we can work with this small biopsy and also the cytology in order to have a similar analysis?
Yeah, so from a machine learning perspective, what we need will be the data, right? So once we have the new data types, what we typically do is a similar process to what I mentioned before for melanoma, right? We had a lung model that we built with annotations. We incorporated new annotations from the melanoma tissue, and then we can learn the model to incorporate that. I think the biggest challenge that we see is, depending on the type of tumor that we're working with, being metastatic, non-metastatic, the number of annotations sometimes can be a little higher. For example, we worked with a gastric cancer, and the resident tissue was so varied that we had to include many more non-tumor annotations into the model, so that in the end, we're really getting a good tumor selection there. But it's something that we have quite a lot of experience in, going from indication to indication or tumor to tumor. And our pathology and data team, as I mentioned, always work very closely together to make sure that we get there in short order.
So at the end of the day, with a lot of samples, I believe that you will be able also to work on smaller samples.
Exactly. And again, right, the advantage of using these deep learning models is that the model is always learning. So if we have a tumor model for lung, for melanoma, that works really well today, comes tomorrow, we have some new tissue and the model is having a little bit of issue, it's not like we have to start all over. We just take the existing model, we feed new information into it, and we can quickly train that model and increase that performance again. So that's the advantage of using deep learning. The model is always learning. There's no flat curve, right? The more data you expose to it, the better the model gets.
So we have another question that's actually, there's two of them, and I'm going to try and merge them because they're both interesting. And I think this goes to both you, Paulo, and Alan. And it says, can you incorporate additional driver genes into this model development? So in other words, you have BRAF, what other genes would you add into this Paulo to make it even more compelling? Like, is there a panel that you would envision? And, yeah, the other side of this question is, what additional slides would you need to do that? And I think that's kind of like, what numbers of slides would you need to incorporate into the model design? Does that make sense? Or
Absolutely, yes. At this moment, the other gene that are interesting for melanoma are NRAS, the c-KIT. And surely, at the moment, we have specific drug also for the NRAS mutated c-KIT. For instance, the MEK inhibitor for NRAS mutated, c-KIT inhibitor for the c-KIT mutated patients. But I believe this is also something that we can add, Alan. I believe that with samples, surely we can have the same model that can help us.
awesome.
Yeah, and from our perspective, yes, we can incorporate. The way that we typically build these models is, again, we go through phases. First model is just doing the proof of concept, make sure that the signature and the signal is there, and then we expand on that to show sort of the levels of accuracy that you saw today. We talked about BRAF today. We have worked with a number of different markers for other tumor types, and we're excited to explore the different markers, different biomarkers that Paolo just mentioned for melanoma. So we would do separate development for each one of those, and then once each one of the development is done and the model is built, we can join those models. So again, going back to the first question, can you run it all together? Yes, once the models are done, we can run it all into a single sequence and get the results at the end. So it becomes a single step.
I think that's it, everybody. Thank you so much for your time. And Paolo, a special thanks to you for all your support and collaboration. Really appreciate your time. Alan, also thank you for doing a great job. And I wish everybody a great day and a wonderful evening, and look forward to connecting with you all soon. And if you have any questions, you can feel free to reach out to us at, well, either myself directly or info@bioaihealth.com. And if you have additional questions for Paolo, we can provide his details as well.
Thank you, everybody, for your time. Appreciate it. Paolo, it's been a pleasure. We'll be in touch.
Thank you for involving me, and it was very nice to be here today with you and discuss about this very, very interesting data. Thank you so much.
Take care,
Thanks,
Paolo.
everyone, for your time. Bye-bye.
Bye-bye.